Hello friends, welcome back to our YouTube channel Doctor's Corner. Today we are going to discuss about erythropoiesis. Erythropoiesis means the process of formation of RBCs, that is the red blood cell or erythrocytes. So how the red blood cells or erythrocytes are formed, the process of production of that RBCs is known as erythropoiesis. So before going in detail about that topic of erythropoiesis, we briefly will discuss about the structure of a RBC. So RBC is basically a biconcave circular disc. Okay, if you see from top, it will appear like as a circular disc having a diameter of around 7.8 microns. You can take it as 7 to 8 micrometer. Okay, but if you see it from this side, it will appear as if it is a biconcave dumbbell shaped structure like this. Okay, as I said you, the diameter is around 7.8 micron. And it is thickened at the periphery and having a thickness of around 2.5 micrometer, and in the center it is around 1 micrometer. Okay, so this is from this side. Okay, to make this point more clear, I'll just draw this thing. So consider this as a cut section of an RBC. Okay, this is an RBC and this is a cut section. So from side it will appear as if it's a dumbbell shaped structure, and if we see from the top, it will appear as a circular disc with the central harrow like a button. Okay, if you see from the top, it will be like this, and from the side it is like this, and the total volume of an RBC is around 90 to 95 cubic microns, a volume of a RBC. So these are the basic dimensions of an RBC, and this RBC is a anucleated cell. It doesn't have a nucleus and mitochondria. Also, the RBC contains a very important protein known as hemoglobin. Okay, around 34% concentration of the RBCs will be hemoglobin. We'll discuss about that hemoglobin concentration and all. So one more important thing about the morphology, cell membrane. So this cell membrane of an RBC, so basically there are many proteins. It is a trilaminar structure. On the surface of the cell membrane there are many proteins, but I'll just tell you the important ones. So just inside this there is one protein, that is structural protein known as pectrin. And as on the surface of the RBC there are other proteins like band 3, glycophorin, A, B, C and all. So if you don't remember all the names of the structural protein, it's okay, but try to remember this very important protein pectrin. And one more is the ankyrin. Okay, there is a very clinical significance about this protein because they help in maintaining the shape of an RBC, that is the biconcave shape. Okay, so there are certain advantages of this biconcave shape of an RBC. So what are the advantages of this biconcave shape of an RBC are, because you can see it's not a complete a circular disc or a spherical structure, it's a biconcave structure. So certain advantages will be there for that biconcave structure. One advantage is it can pass through the narrow capillaries. So in our body there are certain smaller narrow capillaries will be there having a diameter of around 3.5 micron. So our RBCs can pass even such a small or narrow capillaries of 3.5 micrometer also. Okay, but that is the one advantage. Another one is, because of this biconcave shape, the surface area of the RBC is more as compared to the volume of an RBC. Okay, so because of this greater surface area, as you know, the main function of an RBC is transport of gases, that is transport of oxygen and carbon dioxide from the lungs to the different cells and tissues. It supplies the oxygen. So for the diffusion of the gases from the RBC to the tissues and again from the tissues to the RBCs, so this greater surface area helps in diffusion of the gases. Okay, so that will be a bit easy. Then thirdly, because of this larger surface area, when RBC can withstand certain osmotic pressure, that is its membrane won't burst if it is kept in a hypotonic solution. So because of this larger surface area, it has certain advantages, that is biconcave shape. structure. Okay, now to maintain that shape, on the cell membrane there are certain proteins, okay, membrane proteins or structural proteins, that is pectin and ankyrin. So these are very important. There are certain clinical significance. So sometimes hereditary, genetically these proteins will be absent. So in that condition is called as hereditary spherocytosis or hereditary elliptocytosis. So what actually happens is, due to the deficiency of these structural proteins which maintain the shape of an RBC, so because of the deficiency of this, instead of this typical biconcave structure, it will be like a globe, okay, it will is known as sphere, it is like a sphere. So that's why it is known as hereditary spherocytosis, okay, and also hereditary elliptocytosis. We'll discuss that if time permits in some other videos. Right now we'll come to our main topic, erythropoiesis. After having this basic introduction about the cell membrane and cell dimensions, we will discuss now the stages of an erythropoiesis. Okay, so what are the various stages of an erythropoiesis, and before that, site of an erythropoiesis. Site of an erythropoiesis we'll discuss. So site means where exactly the RBCs or erythrocytes are formed in our body, what are the, in which organ, okay. So we'll consider this from right from the intrauterine life, that is just before the birth. So firstly, it will appear in the yolk sac. It will appear in the yolk sac by around second to third week of intrauterine life. Remember, I am speaking the baby is not yet born, it's still in the womb of a mother, that is, it is in the intrauterine life. So still in the intrauterine life, the RBC first production start, that is in the first 2 months. So it will be in the yolk sac, and that stage it is known as mesoblastic stage. Mesoblastic stage, okay. Then around third month of intrauterine life, around third month of the intrauterine life, so the RBC production starts in very important organ, that is the liver, and sometimes also in the spleen, okay. So this stage it is known as hepatic stage. Liver, that's why it is known as hepatic stage. So mesoblastic stage, hepatic stage. Then around 4th month onwards, the RBC production starts in the bone marrow, okay, bone marrow, and ultimately by the end of the normal gestational period, around 8th month, okay, just 1 month before the the delivery, that is the total duration of the pregnancy roughly is around 9 months. So by the end of 8th month, this bone marrow is the principal site of the production of the RBCs, and this stage it is known as myeloid stage. Myeloid stage, okay. So right now what we are discussing is just before birth. So in the womb of a mother, that is in the intrauterine life itself, the production of the RBC starts, okay. So at the second or third week of the intrauterine life, as early as second and third week, this will start, and it will start first in the yolk sac. So this period it is known as mesoblastic stage. Then around 3rd month it will start in the liver and also in the spleen, and this stage it is known as hepatic stage, and finally around 4th month the production starts from the bone marrow, and ultimately by the end of the 8th month the bone marrow will be the main site, that is at birth the process of erythropoiesis or the production takes place only in bone marrow. It will stop in this liver and the other structures, okay. So at birth the bone marrow is the principal organ or the structure in which the erythropoiesis takes place, okay. Now once the baby is born, as the bone marrow will produce the blood cells and erythropoiesis, that is the RBCs also. As the baby grows up, so by the age of puberty, that is around 18 to 20 years, few of the long bones, the bone marrow will become yellow bone marrow. So I'll just like to highlight this point here. What is red bone marrow and what is yellow bone marrow, okay. Red bone marrow as well as the yellow bone marrow. Red bone marrow is known as active bone marrow, and yellow bone marrow it is known as inactive bone marrow. That is, active bone marrow means the active cells, that is the cells will multiply and differentiate and having a growth in red bone marrow, whereas yellow bone marrow such growth and differentiations of the cells is not seen. So at birth basically all bones will be the red bone marrow, that is in all the bones the production of and differentiation of the cells takes place. What are the stages we'll discuss now. Before that, this one. So but around the age of puberty, what happens is the long bones, bone marrow of the long bone will become the yellow bone marrow, that is the process of erythropoiesis doesn't take place in this bone marrow. So after the age of 20 years, after the age of the 20 years, so it is mainly in the axial bone marrow. Okay, axial bones, bone marrow. Axial bones means bones which are present in the axial system of the skeleton, that is a skull, sternum, ribs, vertebra, hip bone. So the bone marrow of this will produce the RBCs or erythropoiesis, and the peripheral long bones basically they consist of the yellow bone marrow, except for the proximal end of the long bones like the femur or humerus or tibia. Okay, rest all long bones will have the yellow bone marrow, and this is erythropoiesis doesn't take place, and it will take only in the axial bones, okay, which has the red bone marrow. That is, after the age of 20 years, 50% of the total bone marrow will be red and 50% will be the yellow bone marrow. Yellow bone marrow mainly in the long bones and red bone marrow in the axial skeleton. Okay. Now, sometimes what happen is, due to some pathological condition, for example, aplastic anemia, in which there is no production of the blood cells or erythropoiesis in the bone marrow, then the production of the RBCs takes place from some other sites like liver or spleen, okay, or some lymphoid tissues. Then that is known as extramedullary hematopoiesis. Okay, that is a pathological condition. So sometimes it takes place. Normally, apart from the bone marrow, some other sites does not takes place. Okay, very rarely. So some in pathological conditions. Then even this yellow bone marrow can become the red bone marrow to produce the blood cells, okay, that is known as, and in other tissue that it is known as extramedullary hematopoiesis. Okay, so this is the site where exactly the production of the blood cells, or in particular erythropoiesis or the production of the RBC takes place. Now we'll come to the stages of the production of erythropoiesis. Okay, so in the theory exams of various universities, several times this questions has been asked as a 10 marks long essay question. Describe the stages of erythropoiesis and add a note on regulation of erythropoiesis. Okay, so stages and regulation of erythropoiesis, very important theory question, and clinically also you are supposed to know these things. So in the subject physiology of first year MBBS, this has been asked several times. So we'll discuss about that various stages of the erythropoiesis now, and also briefly about the regulation, how it is regulated. Okay, so first we'll discuss about the stages of the erythropoiesis. I hope the concept is clear by this point. So consider this as the bone marrow. Okay, so what I am now highlighting here is, this is the bone marrow house. Okay, so this is the bone marrow. So this is the bone marrow. So in this bone marrow, basically all these blood cells, that is either red blood cells or other cells like WBCs and platelets, they are derived from basic one cell, and that basic one cell, it is known as stem cells, and that stem cell, it is known as pluripotent hematopoietic stem cell, PHSC. Okay, pluripotential hematopoietic stem cells, pluripotent hematopoietic stem cells. They are uncommitted stem cells, hematopoietic stem cells. Okay, so from this larger cell, basically in the bone marrow, now what we are discussing, this is the bone marrow. Okay, so in the bone marrow, what happens is, there is a production of all the blood cells, and mainly the now what we're discussing about the RBCs. Okay, so this is a very immature cell or the stem cells having a large nucleus and the big size cell. This is known as pluripotent hematopoietic stem cells. Okay, so from this pluripotent hematopoietic stem cells, two types of cell lineages will originate. Two types of the cell lineages will originate. That is, one is common lymphoid stem cells, and also common myeloid stem cells. That is, they will differentiate and grow into common lymphoid stem cell and a common myeloid stem cells. So these are basically the stem cells and uncommitted stem cells. So from this lymphoid stem cells, basically B lymphocytes and T lymphocyte, which are the types of an WBC, B lymphocyte and T lymphocyte, which are types of a white blood corpuscle that is developed. Okay, now from this myeloid stem cells, apart from this B and T lymphocytes and the natural killer cell, rest all the blood cells like platelets, RBCs and the different granulocytes takes origin, monocytes. Okay, so there are different series. So from this myeloid stem cells, there are many things, but for our particular topic, in our cell of interest is colony forming unit erythroid series. Okay, E. So there will be many such series from the myeloid stem cells. For example, colony forming unit megakaryocytes, colony forming unit GM, that is granulocyte and monocyte, and so. Okay, but we'll just discuss about this colony forming unit erythrocyte. Now these are committed cells. All are immature cells that will ultimately develop into blood cells. So I'll just draw here a blood vessel. Okay, so in the blood vessel, that is in the circulation, various cells will be there. So here mature RBC will be there. Okay, so all this mature RBCs, that is in around the normal quantity is around 5 million cells. Okay, 5 million cells per cubic millimeter of blood. That is, per cubic millimeter of blood there are 5 million RBCs on an average. Okay, so that 5 million cells which are present in the per cubic millimeter of the blood that are produced by this pluripotent hematopoietic stem cell, these are uncommitted stem cells, and ultimately this colony forming unit, they will form this RBCs by undergoing the various stages. Okay, so now there are certain characteristics of these stem cells. What are the characteristics of these stem cells? So stem cell, first thing is, has a property of self-replication. That is, they can multiply their own number. Okay, then secondly, they can have a property of growth and differentiation. That is, they will grow and differentiate into the mature cells. That is the characteristic of the stem cells. So these are the stem cells, these are the stem cells. Now these are the committed cells. That is, they will grow. For example, colony forming unit erythroid series. So if you grow this type of immature cells, then they will, in a culture media, then they will grow only in erythrocytes. Okay, and if you grow this colony forming unit megakaryocytes in a culture media, then they will develop into megakaryocytes. Okay, so ultimately these are the main committed cells, these are uncommitted cells. Now from this colony forming unit erythroid, the first cell will be proerythroblast. The first cell will be proerythroblast. So I am just erasing all the things here from this colony forming unit. So first cell in the stages of erythropoiesis will be proerythroblast. Okay, proerythroblast. So proerythroblast, basically it's a very larger cell having a diameter of around 15 to 20 microns. Okay, a very big cell, 20 micron, and a larger nucleus. 15 to 20 microns and a larger nucleus. Okay, now this is known as proerythroblast. That is the first cell, it is the immature cell. Ultimately this proerythroblast develops into the RBCs in 7 days. So what are the other stages we are going to discuss? Now after that, the second stage will be basophilic erythroblast. Erythroblast means immature erythrocyte, that is the blast cell. Basophilic erythroblast. So this basophilic erythroblast have the cell size is bit less as compared to this proerythroblast. It is instead of 15 to 20 microns, it will be around 20 to 16 microns in size, diameter. Okay, at the nucleus is condensed, that is the size will be condensed and having a this one chromatin will be fine here, and in this cytoplasm there will be bluish color seen when stained. Okay, so that's why it is known as basophilic erythroblast. Okay, why it is basophilic is because if you remember the central dogma of protein synthesis, that is DNA. In the biology, if you might have studied, DNA forms the RNA and RNA ultimately forms the proteins. Okay, so this process of formation of RNA, messenger RNA from the DNA, that is known as transcription, if you remember the basic biology principles, and from the RNA to the protein synthesis it is known as translation. Okay, so transcription and translation. So here this process will start up, that is this DNA, the genes which are responsible for the formation of hemoglobin. You know, before that in this proerythroblast there will be no hemoglobin. In the mature RBC there will be hemoglobin will be there. Okay, so in the immature RBC there will be no hemoglobin present. So ultimately the cell will designate to produce the more RBCs, more hemoglobin. So this nucleus DNA, they will form for the formation of the protein synthesis, they forms the RNA. Okay, RNA and ribosome. So because of this RNA and ribosomes, this will appear bluish color, that's why it is known as basophilic erythroblast. Okay, then the third stage is polychromatic erythroblast. Polychromatic erythroblast. Okay, again the cell size further decreases. It will be around 10 to 12 microns, and the nucleus still condenses, the nucleus size still decreases, and here again due to the presence of RNA there will be some basophilic material. Also the production of hemoglobin starts here, that is the RNA will develop protein synthesis, that is formation of hemoglobin starts here, and it will give some pinkish appearance. Okay, that's why it is known as polychromatic erythroblast. So the cytoplasmic staining, it has both basophilic as well as the pinkish staining due to the presence of hemoglobin. Okay, that is hemoglobin formation starts first at the stage of polychromatic erythroblast. Then the next stage will be, that is the fourth stage. So the next stage is fourth stage, it is known as orthochromic erythroblast. So this orthochromic erythroblast, the cell size further decreases, it will be around 8 to 10 microns, and the nucleus further condenses. Okay, and the nucleus further condenses, and the basophilic, that is RNA and ribosome material, will be very minimal, and majority of the cytoplasm there will be pinkish appearance because of the hemoglobin. Okay, because of this hemoglobin. So this it is known as orthochromic erythroblast. At this point I would like to highlight one important thing is, so in different textbooks you will see some other terminologies also in the stages of erythropoiesis. This basophilic normoblast is erythroblast, it is also known as early normoblast, and this polychromatic erythroblast, it is also known as intermediate normoblast. Okay, intermediate normoblast, and this orthochromatic or orthochromatic erythroblast, it is also known as late normoblast. So this cells is also known as normoblast. So early, intermediate and late, and hemoglobin first starts appearing in the stage of intermediate normoblast. Okay, now the next stage is reticulocyte. reticulocyte. So remember, all these things are happening in the bone marrow itself. Okay, the cell size is further decreases. Okay, the cell size is further decreases. It will be now around 7 to 8 microns. Okay, almost near to the RBC, just slightly bigger than the RBC. And here important thing what happens is the nucleus is extruded out. Nucleus, then mitochondria is extruded out of the cell, and only the cell will be an anucleated cell, and some mesh like network will be there. That's why it is known as reticulocytes. Okay, so it will be like a reticulum. So this is a reticulocyte. Okay, so now this reticulocyte will enter into the blood circulation. How it enter into in the blood, in the bone marrow? This all stages of development taking place in the bone marrow. In the bone marrow, there are smaller blood vessels known as capillaries, and the type of the capillary, there are different types of capillary, like continuous capillaries, fenestrated capillaries, sinusoidal capillaries. So there will be sinusoidal capillaries in the bone marrow. In that blood vessel, that is, capillaries are the smaller blood vessels. So this reticulocyte will enter into the blood vessel, that is the sinusoidal capillaries, and into the, that is nothing but the blood circulation. Now from this reticulocyte, the final mature RBCs is formed. Okay, and this nuclear material is totally lost, and this RBC will be an anucleated cell having the hemoglobin. Okay, having a diameter of around 7.8 micron. Okay, so just to summarize the stages of the erythropoiesis. So the first immature cell from the colony forming unit erythroid series is erythroblast. Okay, it's a larger cell. Then it will gets converted into basophilic erythroblast, then polychromatic erythroblast, then orthochromatic erythroblast, and reticulocyte, and finally mature RBC. This basophilic erythroblast, also known as early normoblast. Polychromatic, it is known as intermediate normoblast. Orthochromatic, it is also known as late normoblast. So basically, what happens is, to summarize, the cell size, that is the immature cell, it is the proerythroblast around 20 micron. The cell size ultimately decreases. Okay, from 20 micron to around 7 to 8 micron. Okay, so one thing is, remember, in this process cell size decreases. Secondly, nucleus, maturation of the nucleus takes place, and finally the nucleus disappear in the mature RBC. Thirdly, cytoplasmic staining characteristics. First it is basophilic, then due to the presence of the hemoglobin in the intermediate normoblast or polychromatic erythroblast stage, there will be polychromatic, that is both, and finally in this that will be more of the pinkish staining because of the hemoglobin. So cytoplasmic staining characteristics changes. Okay, fourth, cell multiplication. Cell multiplication is mitosis. So this is there will be more multiplication of the cells. Mitosis takes place at this stage, here also, but at this stage of the late normoblast or orthochromatic erythroblast, mitosis stops and cell will only grow. Okay, and there will be no multiplication of the cells takes place. Okay, so these are the stages of erythropoiesis. Then how this erythropoiesis is controlled or regulated. Okay, again time won't permit me to describe in detail about the regulation of the erythropoiesis. I will just tell briefly how this process is regulated. So for that there are certain general factors and certain maturation factors. Okay, certain general factors and maturation factors help in regulation of erythropoiesis. Okay, special maturation factor, general factors and special maturation factors. Okay, see, for the development of this mature cell from this immature cell, there are so many growth inducers will be there, and other differentiation. For the process of differentiation of these cells, certain chemicals like interleukin 3, interleukin 1 and 6, insulin like growth factors, all that are involved. Apart from that, some general factors and some special maturation factors are needed. So in the general factors, the most important chemical or a hormone which is essential for this process of erythropoiesis is, remember, very important, erythropoietin. Remember this chemical, it's an hormone, erythropoietin. So it's secreted in the kidney. From the where exactly in the kidney? Peritubular capillaries. Okay, peritubular capillaries means in the kidneys nephrons are there. In the nephron there are tubules. Okay, Henle's loop, ascending limb and all. So just beside that there are peritubular capillaries. In the endothelial cells of the peritubular capillaries, this erythropoietin hormone is secreted. Okay, and this erythropoietin hormone is essential for the all these changes to take place. Okay, that will stimulate this process of erythropoiesis. Okay, now mainly in the kidney, however a small amount of the erythropoietin is also secreted from the liver also, but main source is from the peritubular capillaries of the kidney. So what are the factors that will control the erythropoietin secretion? There are many such factors, but the most important one is the hypoxia, tissue hypoxia or low oxygen content to the tissues. So whenever there is a low oxygen content to a tissue or hypoxia to the renal tissue, then it will stimulate the secretion of erythropoietin, and erythropoietin help in the process of erythropoiesis. Okay, now apart from this hypoxia, other hormones like thyroid hormone, male sex hormones like testosterone, that is known as androgens, then anterior pituitary hormones like thyroid stimulating hormone, adrenocorticotropic ACTH, adrenocorticotropic hormones, prolactin, that also stimulates the secretion of erythropoietin, whereas estrogen, female sex hormone estrogen, will inhibit the secretion of the erythropoietin. Okay, so that's why the RBC count is more in the males as compared to the females. Okay, so this is one of the general factor, erythropoietin. Then for this nucleus maturation or the synthesis of the DNA and all, very important special maturation factors which are required is vitamin B12 and folic acid. Vitamin B12 and folic acid. Okay, so vitamin B12 and folic acid is required for the nuclear maturation. If there is a deficiency of this vitamin B12 and folic acid, this nucleus maturation does not take place, and this cell will be in the immature state, that is a normal RBC does not have a nucleus. So in sometimes what happen is there will be megaloblastic anemia, deficiency of this. Megaloblast means a larger cell, this nucleus maturation does not takes place. Okay, so this vitamin B12 which is absorbed from the GI tract, for the absorption there is one more factor required known as the intrinsic factor of Cassel. Okay, and deficiency of that leads to the pernicious anemia. We will discuss detail about this in the next videos or the coming videos about anemia. Timing, just remember vitamin B12 and folic acid are very much essential for the maturation of the nucleus development or of the RBCs in the process of erythropoiesis. Okay, then other factors like hemoglobin, iron is also required, and apart from that other nutrients are also required. Okay, so this is all about the today's topic of erythropoiesis. If you like our lecture, please share and comment about this video. Thank you.